Okay, so good morning to all of you. So I propose to start, and we are pleased today to welcome Sam Mugford. Sam is coming from John Innes Centre in UK, Norwich. And Sam is a full researcher in John Innes and is working in the group of Saskia Haugenhout, all right? So a few words about Sam, just a few words, don't worry. So Sam is working on the interaction of plant and their aphid pest, and we have some picture here. And you are interested how the plant can, in fact, respond to this aphid and how aphid can manipulate the plant in order to improve their virulence, if I can say. And just concerning your life science, in fact. You work in a PhD, you did your PhD in Oxford, yeah. Right? And then you moved directly to John Innes Centre for 2 postdocs, if I'm correct. And the first one, and maybe the second one also, was more on plant metabolism. And then you integrate the group of Saskia. And so we are pleased to listen to you, and thanks for coming. And just to say that Sam is here today because this is the context of the graduate school. Sam was invited by the graduate school in order to give some courses to the student, the Master 2 student, and also for the roundtable. And tomorrow we have another guest, Cedric Fechot, who will come from US and talking about transposer. So you are invited to assist also to this seminar. So thanks, Sam.
Great. Thank you, Jean-Philippe. And yeah, hello everybody. Thank you very much for coming, and thank you for the invitation. It's a really great place to visit. This is the most spectacular seminar room I've spoken in, and yeah, it's really quite something. So yeah, as Jean-Philippe said, this is Mysis persicae, or Mrs. persicae, as we sometimes call her. This is the green peach aphid, and this is our favorite model system. Phenomenal crop pests, also very beautiful and really fascinating biology. As Jean-Philippe said, I work at the John Innes Centre in Norwich, which is in the east of England, on the little bit that sticks out. Yeah, my pointer is a little bit laggy. Beautiful medieval city, well worth a visit. Great place to come and do science. As many of you probably know, we also have the Sainsbury laboratory next door to us, and I think there's a few familiar faces here who've probably experienced that place, and there's a lot of excellent science that happens there in crop breeding, plant biotechnology. Fundamental molecular mechanisms really is our focus, and that goes across a range of plant and microbial sciences. And within Saskia's lab, at least, and a couple of other labs, we're looking at plant-insect interactions, and here's a couple of our favorite insects that we work on. And this is Saskia Haugenhout's research group, which I'm part of. This is Saskia who runs the group. And the work I'm going to talk about today is work that I've done, but also a lot of work from 2 postdocs, Matteo Gravino and Chuin Liu, both of whom are going to be on the job market soon. So they're both incredible scientists, so keep an eye out for them. And most of the work in the group is focused on the interactions of sap-sucking insect pests. These are insects from the Hemipteran order, which feed by tapping into the plant vasculature and feeding from the sap. Big crop pests, mostly not because of the damage they do to the plants, but because of the pathogens that they transmit. And aphids are the major vector of plant viruses. More than half of plant viruses are aphid transmitted, but there are also a lot of bacterial pathogens that are transmitted by insects, and that forms a second strand of the research in the Haugenhout group, which is looking at the phytoplasma bacteria. These are intracellular bacteria that are transmitted by leafhopper vectors, but I'm not going to talk about those today. I'm going to talk about the molecular mechanisms of the interactions between aphids and their host plants. And in Saskia's lab, we look at that question really through the lens of molecular plant microbe interactions, what we've learned from plant pathogen interactions at a molecular level. We're using that knowledge to understand how insects interact with plants, and these diagrams kind of show the model that we're working with. This is not to scale. The aphid would be rather bigger than the plant cells here, but it shows how the aphid mouthparts, specialized piercing, sucking mouthparts called stylets, penetrate the plant but navigate intercellularly. So they avoid damaging cells and navigate to find the sieve element of the phloem cell where they feed from the nutrient-rich sap. But on the way, they're piercing and probing the epidermal mesophyll cells and delivering saliva into those cells, which contains effector proteins that interact with the plant immune system. And the model we have is that plants will perceive conserved aphid signal molecules via cell surface receptors, and the aphid effector proteins suppress that immune signaling, preventing the plant from mounting an effective defense, and so the aphids can colonize. And I'm going to talk about two stories about the molecular mechanisms here and what we've learned about this. So aphids belong to the Hemipteran order of insects, which include a lot of sap-sucking herbivorous insects. In fact, 90% of hemipterans are herbivorous, with the exception of some blood feeders, the assassin bugs, the kissing bugs, and the bed bugs, all pretty unpleasant things, some of which transmit important human pathogens. It's interesting to note that these have evolved from sap-sucking insect pests. And I think there are some interesting questions about how they've used their adaptations to colonize mammalian hosts. But importantly, for us, this group includes the aphids, which are a very diverse group of about 5,000 species, as well as whiteflies, leafhoppers, psyllids, and plant hoppers, which also include a lot of important pests. And Mysis persicae are our favorite insect model system, is pretty amazing in that it's a polyphagous generalist insect. This is really the exception for herbivorous insects. The vast majority of herbivorous insects are specialists, which will only feed from one species or one genus of host plant and will die if you try and feed them on another, whereas Mysis can feed from more than 400 different plant species, so far as we know, from at least 40 different plant families across the flowering plant taxa, and that includes a lot of important crop pests in which it transmits viruses and is a problem, but also some of our favorite model organisms, Arabidopsis thaliana and Nicotiana benthamiana, which is really nice. And this just shows some of the damage that's done to sugar beets by and wheat by viruses that are transmitted by Mysis. We really want to understand how Mysis suppresses plant immunity with the aim of subverting that, so that we can enable plants to mount an effective response to create aphid-resistant plants. And if the plants are resistant to aphid, then the spread of viruses is no longer a problem. But first, we need to understand those molecular mechanisms. Now, the life cycle of aphids is really fascinating because they have a largely asexual reproductive cycle. So during the summer months in particular, they give birth to live young clonally. So there's no meiosis involved. They are clones of their mother. They're all female. They're born live and genetically identical. And because no mating is required, they can be born pregnant. And in fact, they telescope the generations so they can be born pregnant with their own great granddaughters. And this enables incredibly fast reproduction. When the conditions are right, you'll notice one day you'll go in your garden and your rose bush is covered in aphids where there were none a week ago. And that's really facilitated by this. And of course, we know asexuality has its problems because you end up with a dead end, you accumulate mutations and a virus comes along and wipes out the entire population because you can't shuffle your genes. But they keep the best of both worlds. So they also have a sexual reproductive cycle, which is typically seasonally regulated. So towards the end of the summer, when the temperatures drop and the days get shorter, that induces a developmental change. They'll produce sexual males and females and go through the sexual reproductive cycle, shuffle their genes, get ready with a diverse population for the next season, which all of this really makes them a perfect pest and a farmer's enemy. So I think a lot of you know about plant immunity. And so this is the framework that we're looking at our problem. there are plant cells have cell surface receptors, pattern recognition receptors, which perceive conserved molecular signatures, pathogen-associated molecular patterns. We're thinking about herbivore-associated molecular patterns. And we know that a crushed up aphid spread on a plant will elicit an immune response. We don't know exactly what the molecule is, but we do know that there is a cell surface receptor, which requires the BAC1 co-receptor. So everything points towards a receptor kinase mediated perception. And the second line of this model is of, the classic Jones and Dangle zigzag model is that the pathogens or aphids secrete effector proteins that interact with this process, suppress the PAMP-triggered immune response, and in turn plants evolve the effector-triggered immunity with intracellular receptors that recognize effectors and mount a stronger second line of defense. And what we also know from more recent work is that there's significant crosstalk between these pathways and that ETI and PTI responses can mutually potentiate each other. And I think this is quite a rich area for understanding the molecular mechanisms of those interactions. So we've been thinking, how do aphids fit into this pathway? Well, it's worth knowing a little bit more about how aphids feed. I'm not sure if I can start this. This is a method we use to study aphid feeding. It's called the electropenetration graph technique. It seems a little bit unlikely, but what we do is we glue an electrode onto the back of the aphid. You can see here, the other electrode, we stick it into the soil, and then we measure the voltage across the system, which you can see in this graph below, which is tracking the video. I want to skip ahead in this video, which means I think I need to change my highlighter. No. I need to turn my laser pointer off to do this. There we go. I think if we go along here, you can see that there's a lot of stuff happening in the video. What I can tell you, these little potential drops here, these little square waves, these represent the probing of individual mesophyll cells within the leaf. And that's the phase in which the aphid is delivering saliva directly into those cells. And we now know that that involves effector proteins that are suppressing plant immunity. And that longer potential drop with the fuzzy bottom, that's a phloem feeding phase. There we go. That's where the aphid's tapped into the phloem and is sucking out the sugary sap. So this is a really useful method. We can quantify these different feeding phases and see what the effects of mutations in plant immunity are. And it's really helped us understand that. And this is what really this guy, Freddie Chillingi, who invented the EPG technique, he told us more about how aphids feed than anyone else. And the experiments he did to deduce what's happening during these different phases are quite incredible and dedicated, but I won't go into that. But suffice to say, we can see from these that aphids are making these probing events. We now know they're delivering saliva into those cells and then feeding from the phloem. And so plants are able to perceive this attack, and we know that the aphid feeding is a very stealthy technique compared to the chewing insects. And we can actually have a look at how the plant does respond using the calcium sensor protein, the GCAM, to monitor the intracellular calcium burst in response to the presence of the aphid. And this is a video of a detached leaf with an aphid on it that you can see in a shadow, sped up a bit, and then it finds a feeding spot and there's a little calcium burst there, but it doesn't last and it's quickly shut down. And we think, well, we can show that in comparison to a slightly more destructive pest. Here we've got a thrips, which kind of scrape away at the surface of the leaf. And when he arrives, there you go, doing a lot of damage, and the leaf's just lighting up and going crazy. So the much reduced response to the aphid is in part due to the fact that it's a very stealthy feeder. The stylet is not doing damage, it's wiggling around, but also it's the delivery of the effector proteins and the time scale of that calcium burst, which that's running over about 5 minutes. That matches the time scale over the probing phase. This is the time at which the aphid is probing those mesophyll cells. And then when that calcium burst has been damped down, that's when it's reaching the phloem. So we think the aphid might be actually monitoring the status of the defense system and then not feeding until it's satisfied that the defenses are activated. So how do aphids evade plant defenses? And that's where we think, yes, we're looking for what are these effector proteins. So, you know, we've gathered that the aphids are perceived by cell surface receptors, but they're secreting effectors that are suppressing those immune responses. So we're set about trying to find out what these were. It's not so straightforward. We don't know so much about aphid effectors as we do about microbial pathogen effectors. So bear with me, this should come back. We set about a number of ways in which to identify candidate effectors. And one is, well, let's have a look in aphid saliva to see what's there, which isn't entirely straightforward, but we can in fact collect aphid saliva because we can induce them to feed from an artificial diet. There's my mouse. I need to put the laser pointer back on for those at home. So we set up this system with there it is, a Petri dish with an artificial diet, which is basically a sucrose solution. And on the other half of the Petri dish, we stretch parafilm across it, stick the two together, put the aphids on top of the parafilm and put it on the artificial diet, and they will feed through the parafilm, stick their stylets through, suck up the solution, but deliver their saliva into the liquid, which we can then harvest, concentrate, stick it in a mass spectrometer, and we've identified about 200 proteins that are present in the aphid saliva, some of which look like they have metabolic functions, proteases and glycosyltransferases, and others of unknown function. Also, we've looked at the tissue-specific expression pattern. So we've dissected aphids, which is no small, well, it is a very small feat. In fact, it's literally extremely small. George Seddon-Roberts, who's doing his PhD in the lab, has become very skilled at this, and he's generated RNA-seq data from different tissues, including the salivary glands. And in fact, he got invited to present a radio program on BBC Radio 4. It's a program called Wild Inside, which normally deals with autopsies of dead zoo animals. And they talk about the biology and physiology of these animals. And he recorded an episode of this where he dissected an aphid and talked through aphid biology. So if you're interested in learning more about aphids, I recommend that program. I think it's available from outside the UK. And another approach we took was, because Mysis persicae, being extremely polyphagous, feeds from lots of host plants, we reasoned that part of this adaptation might be the ability to tailor the suite of effectors that it's delivering into different host plant species to meet the demands of different immune systems. So we looked at the transcriptomes of the aphids feeding from 9 different host plant species from 5 different families, identified about 2,500 transcripts that show host-responsive expression. And then these 3 lines of inquiry basically were able to narrow down on a list of candidates that look like they might be the sorts of things that interact with plant immunity. And I'm going to talk about 2 of those. And one is my favorite, and I've done a lot of work with this protein, which we call MP10. So this came out of a screen where we took some candidates, expressed them heterologously in plants, and looked for immune modulatory effects. And we found that MP10 can suppress PAMP-triggered immune responses. So here we're looking at the reactive oxygen species burst in response to a range of different PAMPs, including on the right, aphid extract, so a ground up solution from aphids, which elicits a reactive oxygen species burst. But when MP10 is present, this is reduced, but it also shows suppression of responses to flagellin and oligogalacturonides. So they're a damage-induced signal, plant cell wall components, but not to chitin, which is interesting. So it looks as though MP10 is doing something at a conserved point in the signaling network that's affecting the perception by a whole range of different receptor kinases. MP10 belongs to a protein family called chemosensory proteins. So these are small soluble proteins that have a hydrophobic pocket in the core that are canonically understood to be involved in the perception of chemicals in the environment. They're an arthropod specific gene family found across all insects and other arthropods. And some of them have been shown to have roles in, for example, perception of pheromones or perception of plant volatiles, but in fact, it's becoming clear that they actually have a much wider range of roles beyond chemical perception. And here we think we found one that's taken on a function as an immune suppressant. This phylogeny shows proteins from this family from different hemipteran insects, including a range of aphid species in green, as well as leafhoppers, psyllids, plant hoppers. And MP10 belongs to this clade called CSP4, which has more or less one copy per genome. So this diversified, this whole family diversified at the origin of the sap-feeding hemipteran insects. And we found homologs, orthologs of MP10 from this clade share the plant immune suppressive activity. So it looks like a pretty ancient effector protein, because we're looking back at the divergence of these species about 250 million years ago, which is really the point at which vascular plants were first interacting with sap-feeding insects. So this is something that's been happening for a long time. We don't tend to think of these relationships as so long-lived because of coevolution. So we didn't detect MP10 in the saliva of the aphids from the artificial diet, but we do detect it in plant cells at the feeding sites where aphids have been feeding. So we did this using a specific antibody we raised using immunogold labeling. We took sections of Arabidopsis tissue from the feeding site where aphids have been feeding, and we labeled them with the MP10 antibody and a gold particle secondary. So here we can see mesophyll cells with, this is the track left by the stylet. They secrete a gel around the stylet, and here we can see it between these three mesophyll cells. And under the electron microscope, you can see the pore through the middle through which the stylet has penetrated. And adjacent to this, there's a section of cytoplasm. So these are highly vacuolated cells with the cytoplasm around the edge. And we can see labeling of with the MP10 antibody in the cytoplasm of this cell. And we only see that adjacent to the stylet track. We don't see it in other parts of the leaf or in uninfested leaf. So we see that indeed the aphid is delivering MP10 into the plant cell. And we were thinking about, why don't we detect it in the saliva in the artificial diet? And actually this came from a collaboration with Lynn Margulis in Montpellier. And she had previously identified the structure at the tip of the aphid stylet, the tip of the mouth parts, which she called the acrostyle, which seems to be a binding platform. And she'd found that, in fact, some plant viruses stick to this surface. And this is a protein from cauliflower mosaic virus, which adheres, labeled with GFP, to this stick, so it can be delivered directly from the tip. And with her, we found that MP10 also binds to this structure. So we hypothesized that MP10 and potentially a whole suite of other effectors are sitting on this platform and are only released into the plant cytoplasm under specific conditions. And presumably our artificial diet doesn't meet those parameters. So this is the model that Marilyn came up with, where we have MP10 bound to the acrostyle at the tip of the stylet, and when it penetrates these plant cells, that's where it's released to suppress immunity. So what is MP10 doing in there? We see that it's suppressing a lot of this PAMP-triggered signaling. We look for an interacting partner, and from a yeast-2 hybrid library, we pulled out a deubiquitinating enzyme called AMPH2. This is part of a family of 3 conserved deubiquitinases, AMPH1, 2, and 3. AMPH1 and 3 have previously been shown to have roles in membrane trafficking processes. So typically, they form a part of the escort complex, the escort 3 complex. And interestingly, this is involved in the trafficking of membrane receptors and part of the normal homeostasis of these receptors, which traffics them from the plasma membrane and degrades them in the vacuole. So we thought, oh, this could be how MP10 is regulating membrane receptors. We also found that this is a conserved interaction. So as well as interacting, albeit less weakly, with 1 and 3, MP10 also interacts with AMPH2 orthologs from other plant species. And we also found that the orthologs of MP10 from other insect species including the whitefly leafhopper, 2 leafhopper species, will interact with AMP2 and indeed AMP2 orthologs from other plant species, although not in all. So we verified this interaction by doing co-immunoprecipitations. In plantar, we co-expressed a GFP fusion of AMP2 with flag-tagged MP10. And from total protein extracts, we detected no co-immunoprecipitation. And we thought, come on. And we actually, this took quite a long time to figure out. But of course, we know AMP is not a membrane-bound protein. In fact, it's soluble, it's found throughout the cytoplasm, but it's doing its job as part of a membrane-associated complex. So we thought, well, what if we enrich membrane fractions? So we pulled out membrane fractions, and then we did the immunoprecipitation on the membrane-enriched fractions. And this is a little bit of a complicated one to interpret, but basically in the co-IP, where we pull down GFP AMP2 from membrane fractions, we pull MP10 down with it. So great, there's an interaction. And we've done this in both directions, and we can pull it down from membrane fractions. In fact, what we see when we label the co-IP with the MP10 antibody, we see this band at 70 kilodaltons, where the expected band at about 26 whatever kilodaltons is there, but weak. So there is some high molecular weight complex that is SDS resistant that labels with the MP10 antibody, and we don't quite know what else is in there, but it's an interesting observation. Interestingly, AMP2 isn't, we see that's the expected size of the AMP2, but it doesn't label the 70 kilodalton complex. So although they're interacting, it's a SDS sensitive interaction, but there's something else in this complex. Anyway, so we've verified the interaction. They're interacting at a membrane. We did some FLIM FRET experiments as well. And this is with a GFP tagged MP10 and a M-cherry tagged AMP2. This is just showing a section of cytoplasm where you can see both of them are ubiquitously spread throughout the cytoplasm and co-localized. But when we measure the FLIM FRET and we look at the fluorescence lifetime, we see a drop in the lifetime when we have MP10 and AMP2 present, and also MP10 and AMP1, consistent with an interaction between these proteins. And we don't see this lifetime change in the presence of controls where we have AMP2 with a free GFP or MP10 with a M-cherry Acorin fusion. So it looks reasonably specific. When we map the efficiency of the FRET, that's basically saying where the interaction is happening. This is showing FRET efficiency, and this is showing the GFP intensity. So this is where MP10 is. And here in red and yellow is where the interaction is happening. And in blue, that's where the interaction is not happening. So we can see that there is a specific location, or there are specific locations within the cell where these two proteins are interacting, even though they're co-localized all over the place. And presumably this is a membrane compartment of some sort. We don't know what. So that's interesting. And then as a control to confirm that these FRET signals are indeed due to FRET, we look at a phasal plot, the physics of which is incredibly complicated and I don't get my head around, but the experts tell me that the movement clockwise around this plot from here to here is indicative of a bona fide interaction. So what is MP10 doing to the receptor kinases? Well, we looked at the expression of FLS2 as a GFP fusion, the flagellin receptor, because we know MP10 suppresses this response. We can use this as a model. And this is showing FLS2 GFP on the periphery of a cell, on the cell membrane, and co-localizing with a plasma membrane marker, as expected. When MP10 is present, we see additional structures appearing. So we start to see FLS2 on intracellular structures. These we thought looked like transvacuolar cytoplasmic strands. So this is consistent with FLS2 appearing on the tonoplast membrane. And here you can see clearly where the plasma membrane marker is on the outside, we're seeing FLS2 on the inside. And when we did this experiment with a marker for the tonoplast membrane, the SUC4 transporter, you can see co-localization of FLS2 and SUC4 when MP10 is present. So it looks like MP10 is causing internalization of the receptor kinases, drawing them away from the plasma membrane, which makes sense because that's going to render the cell insensitive to the extracellular signals. And we presume that this involves the AMPS proteins because they are involved in membrane trafficking processes. And this graph shows an experiment where we have used a dominant negative form of AMPS, which has an active site mutation, lacks deubiquitinase activity. So we overexpress this in Nicotiana cells to essentially inhibit the endogenous AMPS functions. And the first thing to see is when we express MP10, we're measuring here the reactive oxygen species burst in response to flagellin. So we see a suppression of PAMP-triggered immunity. And then when we express the dominant negative mutant of AMPS2, we see a similar suppression of ROS burst. So AMPS2 is clearly doing something similar when we inhibit it, or rather AMPS2 is required for the normal ROS burst. In fact, this we anticipate will suppress the activity of all three AMPS proteins, so there is involvement of AMPS proteins in general. And when we co-express with MP10, we don't see any additional suppression, so it looks like they're operating together in the same pathway. And then as a control, we've overexpressed the wild-type AMPS, and this doesn't have any effect. So it looks like MP10 is acting through AMPS proteins to relocate the receptor kinases. So from the membrane fraction co-immunoprecipitation, we also did mass spectrometry, and we identified, in addition to MP10 and AMPS2 pulling each other down, a number, I think about 19 different receptor-like kinases. So it looks like it's an effect that's pulling, these two are interacting in a complex associated with a membrane that involves a lot of different receptor-like kinases, one of which we thought was particularly interesting, Feronia. This is a receptor kinase with a wide variety of different roles, one of which is as a regulator of other receptor kinases. So we're not sure yet what the role is. We also looked at the abundance of these receptor kinases, at least a couple of them, and we see when MP10 is present on the right, we see a reduced abundance of the receptor kinase. So this is a Feronia red fluorescent protein, and you can see the band, the major band, really disappearing when MP10 is present. And similarly, we did this with FLS2, and we see a substantial reduction in the amount of FLS2, which is consistent with promoting degradation of the receptor. So this is how we think MP10, the aphid, is selectively delivering MP10 into host plant cytoplasm. It's acting through AMPS proteins, and it's targeting receptor kinases of a wide variety of types for internalization, degradation, rendering the plant insensitive to PAMPs. We've been thinking of this as a local anesthetic, much in the way that a mosquito stops you feeling from it biting. The aphid is stopping the plant from perceiving its attack. So how am I doing for time?
10 minutes or more.
Okay, well, I've got a second effector protein that we're interested in that I'm also going to talk about, and this is one that shows differential expression on different plant hosts. I'm going to go a little bit quickly through this, but essentially we found when we looked at transcripts that respond to different host plants, we found enrichment for a family of cysteine proteases called cathepsin Bs, which all show coordinated expression. We also detect these proteins in the saliva of aphids feeding on an artificial diet, and they belong to quite an expanded clade that's expanded in quite recent history since the divergence of Mysis from its close relative species. So this is looking like a more evolutionarily dynamic story. And when we overexpress these cathepsins in planta, we see an enhancement of aphid performance. So they're creating an environment which is more favorable for the aphids, presumably through suppressing immunity. And interestingly, we found when we express these as fluorescent protein fusions, we see accumulation in these structures, puncta or aggregates, which co-localize with a marker of the processing body, P bodies. These are bodies which are involved in the sequestration of inactive mRNAs often during stress treatments where translation is blocked for specific transcripts. And so what the cathepsin is doing there, we were very interested in, but we took a similar approach looking for interacting proteins. In this case, this is work mostly done by Chuen Liu. Chuen did some affinity labeling with TurboID and identified EDS1, which is, for those of you involved in immunity, know is a very central player in plant immunity, especially effector triggered immunity, and showed that the cathepsins would also interact with EDS1 in a yeast-2 hybrid, but not the related partners of EDS1, PAD4, or SAG101, and also detected these interactions by co-immunoprecipitation. So it looks like we have here we have an interaction of an aphid effector with the other branch of plant immune signaling, EDS1 as a key transducer of many of the intracellular receptor mediated processes. As I mentioned, one of the partners of EDS1 is PAD4, and actually, bear with, this has previously been shown to have a role in aphid defense. A mutant of PAD4 has got enhanced susceptibility to mysis persicase, so that was already on our radar. So Chuen then had a look at the effect of cathepsin on the localization of EDS1. So I should have animated this a bit better, but you can see the normal localization of EDS1 is nucleocytoplasmic. It's found throughout the plant cell. But in the presence of cathepsin B6, it shows co-localization, which in these bodies that we presume are P bodies. And again, when we do this experiment showing EDS1 GFP in the presence of a cathepsin B6 without a fluorescent tag, we can see that the EDS1 partially co-localizes with the marker of the P bodies. So it looks as though cathepsin B6 is interacting with EDS1 and dragging it into a P body. So we believe it's sequestering it there. Not a normal function for a P body, but somewhere where the aphid has decided it's going to take a plant immune protein out of action. And indeed, when we look at the effect on aphid performance, as was previously reported, the PAD4 mutant shows enhanced aphid susceptibility. The EDS1 mutant also shows some increased susceptibility to aphids, although somewhat less marked. And then when Chuen looked at the effect of the cathepsin B6 overexpression in these genetic backgrounds, in the wild type, you see the cathepsin overexpression enhances sensitivity to the aphids. But in the EDS1 mutant and the PAD4 mutant, the effect of cathepsin B6 is lost. So both of these proteins are required for the action of this effector. So in summary, we've got two effector proteins that we've looked at out of, I don't know how many are really involved in aphid suppression, acting at different layers of the plant immune system, MP10 acting as a suppressor of PAMP-triggered immunity as a local anesthetic, and cathepsin B6 as a suppressor of effector-triggered immunity. And together, these seem to be doing a good job. And future work to identify actually how these processes interact with each other is high on our priority. But I've got two more slides to add to this story, because you might be asking, well, what triggers effector-triggered immunity in aphids? And in fact, when we express MP10, our PTI suppressor in plants, we see an induction of chlorosis, which looks very much like effector-triggered immunity. And indeed, when we do this in an EDS1 mutant of benthamyana, this chlorosis phenotype is lost. So we think, oh, great, you know, MP10 is suppressing, inducing effector-triggered immunity as well as suppressing PAMP-triggered immunity. But it's not quite that simple, because when we looked at the canonical activity of MP10, the suppression of PAMP-triggered immunity, in the wild type benthamyana, in the absence and presence of MP10, you see this reduction of the reactive oxygen species burst. When we do this in the EDS1 mutant, that's lost. And that's really curious, because this is suggesting that the EDS1 is required to suppress PAMP-triggered immunity. which doesn't make a huge amount of sense, except I think in light of the knowledge now that we know that ETI signaling processes have a regulatory role in regulating PTI, and EDS1 specifically has been shown to be required for ETI potentiation of PTI. So this is suggesting that MP10 might be involved in this pathway to regulate PTI, subverting that pathway in a manner that presumably involves AMPS proteins. So I think this might put the AMPS proteins in this pathway. So AMPS proteins may be involved in the regulation of PTI by ETI. And I think that's something that might be of interest outside of aphid plant interactions. I'm going to stop there. I'd just like to thank the people who did this work. I've got 2 acknowledgement slides because this is the people who are involved in the Cathepsin project with Chuen Liu doing basically all of the work there, but some others involved. And the MP10 work, which was done by myself and Matteo Gravino, and also all of these other people and collaborators. So thank you for your attention.